As part of food science, let us study the module which deals with gelatinization and the process of malting of cereal grains, and see how these two features differ from one cereal to the other. So, looking at the objectives of this module, the main objective is to understand the process of gelatinization and the food products that can be produced as a result of gelatinization, to gain a knowledge of related terminology and its application, to understand the process of malting and its functions in everyday life, to learn the benefits of malting. So, what are the physical characteristics of starch? In order to learn about the actual changes which takes place in the starch molecule as a result of this process of malting, and what is the use of these changes taking place, what are the different changes and stages in this process of malting, and similarly what are the different changes and forms of the starch granule that it undergoes during probably other things like gelation or retrogradation. We have to know the quality of the starch, the content of carbohydrate, the content of fiber, and the amount of protein which is also present. So, based on these factors, the time that starch takes to undergo this change and the temperature at which it is taking place will vary from different types of starches. For example, corn starch, potato starch or wheat starch, all of these will vary. There is no set temperature and period or time period within which the particular change will take place for all cereals. It varies from one to the other. Starch is insoluble in cold water. When starch granules are mixed with water, it forms a suspension, with the starch granules becoming suspended in the liquid. If left undisturbed, the starch does not stay suspended and settles at the bottom of the container. When heated at a temperature range characteristic of that particular starch is applied to the starch and water suspension, the granules swell with water. Corn starch granules swell at 144 to 116 degrees Fahrenheit or 62 to 72 degrees centigrade. When starch granules swell at 136 to 147 degrees Fahrenheit, the potato starch granules swell at 138 to 154 degrees Fahrenheit. as heating continues, the suspension becomes thicker, the viscosity of the mixture increases and it becomes more translucent. This process of heating starch in water or any water based liquid and the swelling of starch granules, which causes thickening and increased viscosity of the mixture, is called gelatinization. If you think of the term gelatin, you can see there are two types of gelatin. You can have gelatin from a vegetable or a vegetarian origin, which is from plants. Gelatin protein can also be got from non-vegetarian origin. It is just a protein and can be got from a animal source also. So, those of you who are not aware of this, you should look at the label of the food before consuming to see whether it is of vegetarian or non-vegetarian origin. But immaterial whether it is vegetarian or non-vegetarian, the property exhibited by the starch, the granule, is the same. That is, it gives that particular texture and consistency to the food product. So, when we say gelatin, students, what comes to your mind? Yes, a lovely dessert, right? You can make jelly, which comes from the gelatin crystals, which you have to get. It is available in the local market, and you can reconstitute it with warm water, and sometimes even some sugar is added along, and then in hot water you reconstitute, you allow it to cool, and then probably you refrigerate it. So, as soon as you do this, you mix the warm water and then you cool it slightly, you will find initially the consistency is very thin. You do not find any change in texture. You can't see any visible change in the structure of the granules which you are using. The gelatin granule, it is just like water. But after placing it probably a half an hour in the chill tray or the freezer and taking it out, you will find that a nice firm thick jelly is formed as a result of the process of gelatinization. So, that is the magic that the granules can do when placed in the water, and okay, that is it. We will go into the detail of this process of gelatinization. Now, the heating must continue beyond the gelatinization temperature to obtain a good gel and avoid a raw starch taste. During usual cooking procedures, most of the starch stays inside the swollen granules. Only a small amount of amylose leaks out of the granules. Excessive stirring or mixing of the starch mixture or heating at high temperatures can cause the swollen granules to break, which can produce a lower quality of food product. So, what is the meaning here? When I say the product, the molecules break, it means the structure of the molecule is not retained, and therefore, it breaks and you get a very watery product and you will not get a jelly formed. Okay. So, all the conditions have to be maintained, the temperature, the concentration of the sugar and the type of gelatin used, all of these, amount of gelatin used, all of this is important for proper gelatinization. Let us look now into the chemistry behind this process of gelatinization. Gelatinization is a process of breakdown the intramolecular bonds of starch molecules in the presence of water and heat, allowing the hydrogen bonding sites, that is the hydroxyl hydrogen and oxygen, to engage or bond more with water. This irreversibly dissolves the starch granule in water. Water acts as a plasticizer, right. So, the structure, if you see, the molecule of the starch that is involved opens up and includes water into its structure, giving you that particular texture or gelatin formation. The process of gelatinization. Three main processes happen to the starch granule. The granule first swells, then the crystal, actual structure of the granule or the double helical structure melts. Okay. So, first is the swelling of the structure with water, then melting of the double helical structure, and finally, the amylose leaching. What is that amylose leaching? That is, amylose contained in the starch granule gets free from that composition into the surrounding mixture. Therefore, it will not hold its structure. During heating, water is first absorbed in the amorphous space of starch, which leads to a swelling phenomenon. Water then enters via amorphous regions, the tightly bound areas of the double helical structures of amylopectin. At these ambient temperatures, these crystalline regions do not allow water to enter. Heat causes such regions to become diffuse. So, the amylose chains begin to dissolve, to separate into amorphous form, and the number of size of the crystalline regions decrease. Under the microscope in polarized light, starch loses its birefringence and its extension cross. So, this is how the structure of the starch granule gradually gets transformed and produces the gelatin. Penetration of water thus increases the randomness in the starch granule structure and causes swelling. Eventually, soluble amylose molecules leach into the surrounding water and the granule structure disintegrates or breaks up. Now, there are different factors which governs the process of gelatinization. Let us see what they are one by one. Temperature. The gelatinization temperature of starch varies with plant type and the amount of water present. Next important point is the pH. Then types and concentration of other ingredients like salt, sugar, fat and protein in the recipe, as well as starch derivatization technology that is used. Some types of unmodified native starches start swelling at 55 degrees centigrade. Other types swell at a higher temperature, that is 85 degrees centigrade. The gelatinization temperature of modified starch depends on, for example, on the degree of cross linking of the amylopectin, the degree of acid treatment, acetylation, that is a biochemical reaction which takes place. Gel temperature can also be modified by genetic manipulation of starch synthase genes. Gelatinization temperature also depends on the amount of damaged starch granules. These will swell faster. Damaged starch can be produced, for example, during the wheat milling process or when drying the starch cake in the starch plant. There is an inverse relationship or correlation between gelatinization temperature and glycemic index. So, what is this term glycemic index? Now, when you want to see the glycemic index of any food, you give a small amount of the food, a measured amount of the food to the subject, and then you see how much the blood glucose level is responding or varying because of this amount of carbohydrate that you have given. So, if you take a food and it has a lot of fiber, for example, it will have a higher or a lower glycemic index. What do you think? Yes, if it has more fiber, it will have a lower glycemic index, because if you recall earlier we said if you take foods rich in fiber, it will tend to bring down the blood glucose level, and therefore you will have the glycemic index of a fiber rich food will be less, whereas the glycemic index of a food which is a more pure carbohydrate or a refined carbohydrate. Can you give me an example of refined carbohydrate? Yes, glucose or any monosaccharide is a refined carbohydrate, and that is directly and quickly absorbed into the bloodstream, and it will also produce a very high and sudden increase in the blood glucose levels, and it is called as a high glycemic index food. So, basically all fiber rich foods will be having a lower glycemic index. Okay. So, let us see now if you are going to gelatinize a starch, what are the benefits? Benefits of gelatinization are: improves the availability of starch for amylase hydrolysis. So, as we know, amylase is an enzyme which acts on the complex carbohydrates. So far we spoke about pectin, amylopectin and glycogen. All these are complex starches, along with cellulose, hemicellulose, mucilages, gums, all of these are complex starch. So, what do you mean by complex? They have to be broken down either by enzyme action or hydrolysis or heat action or acid acting on it to broken down into smaller, simpler sugars like glucose or fructose or mannose, which can be easily absorbed into the bloodstream. So, amylase hydrolysis means breakdown of a complex carbohydrate by the enzyme amylase into smaller, simpler units. So, gelatinization of starch is used constantly in cooking to make the starch digestible or to thicken or bind water, as in the case of a roux sauce or a soup. If you look at the consistency of a sauce, is it running like our rasam or sambar? No, it is a thicker consistency. A soup is a little thinner consistency. So, if you want to alter the consistency, depending on that you have to choose the amount of wheat product which has lesser or more amount of amylopectin and amylase containing flour. If it has a lot of amylase hydrolysis will take place, and it will not be useful for forming a thick product. More of hydrolysis will take place, you will get a more watery product. Next, what is pasting? Pasting occurs with continued heating of the starch mixture beyond gelatinization. Students, if you recall, we said gelatinization is a process by which the starch granules take up water, absorb, they swell. When the heat is there, they sometimes even break, and therefore a gel like matrix is formed. Now, we are talking about pasting, which is heating the same starch mixture, but beyond the temperature of gelatinization, just more than 85 degrees centigrade. The viscosity of the mixture increases as more of the starch granules become swollen. A starch mixture will reach peak viscosity when the number of swollen or intact starch granules is maximized. At this point, the starch mixture is fully pasted. Pasting causes the starch granules to become very swollen and soft, forming a paste. Continued prolonged heating of natural starch mixtures result in decreased viscosity as the granules break and dissolve, and the glucose polymers, that is the starch molecules, become soluble. Pasting is not separate from gelatinization. It is a continuation of the process of gelatinization. Actually, you can say it is the next stage after gelatinization, that is pasting. Most starch mixtures begin to thicken and increase in translucency. What is this term translucency? That is, you know the meaning of opaque, and the opposite is transparent. So, between the two you have a term called translucent. You can understand that. So, that is the consistency we are talking about. Probably when you use corn flour in your house to make a soup, and you can think of that consistency, that is what we call as translucent. It is partly transparent. Translucency at 165 to 190 degrees Fahrenheit. A thickened starch mixture is called a starch paste. If the starch paste is fluid, it is called a sol. An example of the sol is a gravy or sauce. So, a sol can be described as a solid, which is a starch itself, or a solid dispersed in a liquid such as water. If the starch paste is solid, it is a gel. That is right. An example of a gel is a firm pudding. A gel can be described as a liquid, that is water, dispersed in a solid such as starch or a gelling agent. This is exactly what we do when you make a jelly. I had explained earlier. Now, we go to another new term, it is called gelation. Please study the different terms in comparison with each other, only then you will know how to differentiate. Gelatinized starch mixtures exist as salts or gels. All hot starch paste mixtures exhibit the flow properties of salts, but many convert to gels as they cool. Gelation, this is exactly what happened for jelly formation. First they are in a salt state and the liquidish state, and then they form into a gel. Gelation is the formation of a gel. Amylose molecules that leaked out of the starch granules during the process of gelatinization move about in the paste. As the paste cools, amylose molecules move more slowly. When they come into contact with each other, they form what you call hydrogen bonds. Also, it is known as a junction zones. This hydrogen bonding forms a network among the amylose molecules, which results in gelation. Swollen starch granules and the water become trapped in the strong amylose network and a thick gel forms. The concentration of amylose affects the strength of the gel. The higher the amylose concentration, the firmer the gel. The next term we have to look at is retrogradation. Now, gels change with time. As some of the hydrogen bonds holding the amylose network together break with other amylose molecules in the gel, the tendency for amylose molecules to reassociate, come back together in an orderly structure, causes the gel to become dense. Gelatinization disrupts the starch molecules. The following gelation, the amylose molecules reassociate in an orderly structure, forming crystalline aggregates and a gelled texture. The effect of this retrogradation can be felt on the tongue as a gritty texture. Now, do not get confused between all these different terms. We have seen how from gelation, that is a soft sort of a gel, you come to the next stage where you found a thicker gel, and finally now the last one, retrogradation, where you are almost going into a crystalline harder gel which is a little more crystalline in taste, like a gritty texture. So, these are the different terms associated with the process of gelation, and pasting and retrogradation is one of the final stages. Now, we will go into another process known as malting. So, what cereals are usually malted? Can you tell me, students? Yes, only certain cereals which are being tested out the process of malting, and they are mostly barley, but in some other parts also they malt different cereals to brew or distill different type of beverages or other food products which are used typical of different countries in the world. Let us see what is malting. Malting is the process of converting barley or other cereal grains into malt for use in brewing, distilling or in foods, and takes place in a maltings. The place where it is done is called a maltings, sometimes called as a malt house or a malting flow. The cereal is spread out on the malting flow in a layer 8 to 12 centimeters depth. What happens? The process of malting. The malting process starts with the drying of the grains to a moisture content below 14% and then storing for around 6 weeks to become seed, to overcome the seed dormancy. What is seed dormancy? You see all the seeds, every seed does not start to grow. Does the rice you keep in your house grow? No. But sometimes you might find the onion leek you buy and keep, you do not use the small onion, that starts to grow. Why? So, the one which does not grow is called dormant. Only when the condition is right, like the moisture or the temperature is correct, only then it will start to grow. So, that is exactly what they are doing here in malting. When ready, the grain is immersed or steeped in water 2 to 3 times over to absorb moisture and to start to sprout. So, it is allowed to absorb the moisture and then given some time 2 to 3 days to absorb the moisture and to start to sprout. When the grain has a moisture content of around 46%, it is transferred to the malting or germination flow, where it is constantly turned over for around 5 days while it is air dried. The grain at this point is called green malt. The green malt is then kiln dried to the desired color and specification. Malts range in color from very pale through crystal and amber to chocolate or black malts. The sprouted grain is kiln dried by spreading it on a perforated wooden floor. Smoke coming from an roasting fireplace is then used to heat the wooden floor and the sprouted grains. The temperature is usually around 55 degrees centigrade. A typical flow maltings is a long single story building with a floor that slopes slightly from one end to the other end of the building. Flow maltings began to be phased out in the 1940s in favor of pneumatic plants. Here, large industrial fans are used to blow air through the germinating grain beds and to pass hot air through the malt being kilned. Like flow maltings, these pneumatic plants are batch processes, but of considerably greater size, typically 100-ton batches compared with 20-ton batches for flow malting. What are the chemical changes resulting from malting? Malt is germinated cereal grains that have been dried in a process known as malting. The grains are made to germinate by soaking in water and are then halted from germinating further by drying with hot air. By malting grains, the enzymes are developed and that are required to modify the grain starches into sugars, including monosaccharides, glucose, and disaccharides like maltose, and trisaccharides like maltotriose, and higher sugars called maltodextrins. It also develops other enzymes such as proteases, which break down the proteins in the grain into forms that can be used by yeast. Malt also contains small amounts of other sugars such as sucrose and fructose. These are disaccharides, which are not products of starch modification, but were already in the grain. What are the different products of malting? So, what are the different food products that you can make? The malted grain is used to make beer, whiskey, malted milkshakes, malt vinegar, confections such as Maltesers, Whoppers, flavored drinks such as Horlicks, Ovaltine, Milo, ragi malt is also available here, and some baked goods such as malt loaf, bagels, it is a western bread, and rich tea biscuits. Malted grain that has been ground into a coarse meal is known as sweet meal. Various cereals are malted through barley, though barley is the most common. A high protein form of malted barley is often a label listed ingredient in blended flavors typically used in the manufacture of yeast breads and other baked goods. Let us see what are the health and the nutrition benefits of malting. So, first, it is very rich in B vitamins. When you allow a cereal grain to malt, it enhances in the amount of B vitamins. That is, what are the B vitamins I taught you? Thiamine, niacin, riboflavin. Yes. Malt extract may increase the B vitamin content of the beverages. It is used in including folate and vitamin B6. B vitamins are necessary for what? Yes, for carbohydrate, protein and fat in food, which helps to give energy. They also help to regulate and keep your skin healthy. Source of essential amino acids. Yes, so malted products are a good source of essential amino acids. What are the use of essential amino acids? They provide all the essential amino acids required for growth and repair of your tissue, your health. Although some malt extract beverages are not a significant source of protein, they may help provide a small amount of these essential nutrients, boosting your intake. It is very good for the bones. Good nutrition is important for bone health. Some malt extract beverages may not be a significant source of the nutrients your bones need for good health, but they can help boost your intake. In addition to calcium, these drinks may also contain phosphorus and magnesium, also important minerals that help keep your bones healthy and strong. All three minerals make up the primary structure of your bones, while magnesium is also needed to regulate a number of biochemical reactions, hormone conditions, and responsible for mineral metabolism in the body. Therefore, from this module you get to know what is the importance of the processes of malting and the other one gelation of gelatinization of starch, and the different ways in which it can be employed in for cooking different dishes, and also most important the health benefits of germinating and malting, which increase the taste, the digestibility, nutrient availability for the individual. So, make sure to use these different processes and these cereals in your diet in everyday life.